What's up, folks? How's it going? Hopefully you guys can hear me well. More importantly, unable to hear all the goings-ons in my house in the background, which is an ever-evolving scene. Hello, hello. Glad you guys could join us this evening. So, my general plan is just going to be to, I don't know why I have my headset on, but here we are. It's on. So we're just going to keep it on. My general plan is to work through some multiple choice questions using a popular online service that you guys will be familiar with, but I think they make great questions. And using those questions as as sort of a vehicle to teach the content. And as you all have questions, I will come and explain it from my own classroom notes and that sort of thing. And just kind of walk through the units. And that's my plan. You know what? I might have to take these off, I think. All right. There we are. I can hear myself a lot better now, so I don't have my quiet comfort noise canceling headphones on. Not an advertisement. That's just what they're called. Just a quick breakdown of the test. Every year I'll have somebody ask what's going to be on hopefully not what's going to be on the test, but sort of the breakdown. And you've probably seen this. My guess is that you've seen this. I think that it's really important to focus on these last 3 units that we do in the year, and then especially Unit 3, because I think Unit 3 is featured quite a bit on the exam. Unit 4 is also, can be there in force or not. But I think 3, 6, 7, 8 are where you're going to get the most bang for your buck, because I think it's particularly 7 and 8 work their way through all topics, and having a pretty well-versed knowledge of those is going to help. And so let's go ahead and begin. So I'm going to be using Khan Academy that has a lot of good questions, and I encourage my own students to use it as a way to what's the right word that I'm not looking for the word substitute, but add to their own supplement. That's the word I'm looking for. Their own study, and just as a way to practice. We do AP Classroom stuff in our class as well, but I think the AP Classroom questions sometimes are hit or miss, particularly through the year. I think the practice exams were really good, but the AP Classroom questions were not as great. And I think Khan captures the right level of rigor and also bringing the content in. So yeah, definitely love Khan. Let's see if we can do this. All right, there it is. So yeah, shout out to Khan Academy, by the way. They're not paying me. I'm not paying them. But everyone should use their product. If you're planning on taking another AP exam, then use them, because look at all this. It's literally aligned with our units, and great questions and everything. So I'd encourage you to use them. We're just going to go to practice 1 or Unit 1 here, Chemistry of Life, and we're going to just go down here to the just to the unit test and kind of work through that. Though if there is a particular area that you're I really wish we went over that, then yeah, we'll do that. Unit test. We've got 10 to 20 minutes for 10 questions. I don't know how this works. If you can, take it again, because I'm sure the questions are like whatever. All right, here we go. Let's look at this. A team of virologists has isolated a nucleic acid sample from newly discovered virus. In order to classify the virus, they need to figure out whether the viral nucleic acid is DNA or RNA. Which of the following characteristics of the nucleic acid sample would best classify the viral nucleic acid as DNA or RNA? You can set the timer in the corner. Oh, should I? No, I don't like time tests. They're for the birds. So now one of the things I preach at my students, if there are any of my students in this chat, they will confirm this. Go figure out what you're looking for before you come down here, right? Figure out what you're looking for before you come down here, right? And so, which of the following characteristics of the nucleic acid sample would help classify the viral nucleic acid as DNA or RNA? And so I'm automatically thinking, what are the differences? It's the sugar, it's thymine, uracil, right? Single-stranded, double-stranded. Those are the things I'm looking for, right? And so let's go down A, B, C, D. We already have someone said D. Great. Let's look. Nucleic acids of the virus contain nitrogenous bases. They all do, so that's not good. They all have phosphates. They all have covalent bonds. And now here's another test-taking strategy that I know some of you can't even fathom doing. But if A, B, and C are wrong, then D is automatically correct. I don't even have to read it. I know that it's correct because the other three are wrong. You could save time that way. But yeah, it's D. All right. In order to develop antibiotics, scientists need to understand how bacteria grow and reproduce. One factor that is essential for bacteria growth and reproduction is protein synthesis. By understanding how bacteria protein synthesize protein, scientists can attempt to develop antibiotics that block bacterial protein synthesis. So this could easily be Unit 6 question. Which of the following questions will best direct an investigation on how to develop antibiotic that inhibits protein synthesis? So if this was a Unit 6 question, we would think ribosomes, we would think that sort of thing, right? We would think transcription factors, any of that. But since we're in Unit 1, we're probably going to think amino acids. So let's go look. How can the phosphate end of a growing polypeptide chain be blocked? There is no phosphate end to a growing polypeptide chain. So A is not right. How can the hydroxyl end? Again, that's not a thing because the two ends are the carboxyl end and the amino terminus. How can the formation of covalent bonds between amino acids be blocked? This is good, right? We don't even have to look at D because we know that C is the correct answer. But we'll go look at D just to talk about why it's wrong. How can the formation of hydrogen bonds between amino acids? We know that amino acids are monomers, right? And that bonds between two monomers are always covalent bonds. And so this is incorrect. And so C is the correct answer. Good. And if you have questions about how I got one of the answers, or you need me to I guess I can go back I don't know. But if you have those kinds of questions, please ask. Yeah, peptide bonds are a type of covalent bond, correct. Their peptide bonds are specific to the joining of two amino acids. It's like the name of the bond. So all right then, here's some more. In the reaction depicted above, maltose is broken down into two molecules. Which of the following best explains this reaction? All right, so we have a disaccharide or a it's a simple polymer, right? It's breaking down into two monomers. And how do we do that? That's called hydrolysis, right? We add water in order to break. The word hydrolysis literally means water break, right? And so we're going to go find that. The reverse of this reaction will require the addition of water. No. The addition of water molecule cleaves the covalent bonding, holding maltose together. B is the correct answer. Let's go look at C and D and tell why they're wrong, though. The ionic bond between maltose. There are no ionic bonds between monomers. Anytime two monomers are joined together, it is always a covalent bond. The breakdown process requires the release of water. No, you can see that water is not being released here. And so... and I think it's important to underline you've been in this class all year, and so you understand this probably now more than you did when you first came to the class, that this is a reading test, right? And so being able to read the prompt and understand what it's asking is paramount, even more important than content. Because if you can't read, it doesn't matter if you know the content. Which of the following describes the most likely way in which two water molecules will interact? Water molecules are polar, and polar molecules interact with one another by forming hydrogen bonds. Let's go find that. Covalent bond will form? No. Ionic? No. Metallic? No. Hydrogen bond? Yes. See how I did that? I went looking for the answer. I hadn't even read any of this yet, right? I'm not gonna be like, well, I know the hydrogen and the oxygen have a covalent bond, and I'm not like getting in my own head. I'm just pulling the content out and I'm looking at it, right? Keeping things simple. There are 60 questions on the exam, and you have 90 minutes, and so that gives you like a minute and a half per question. And so on a question like this, like you can answer this question in 4 seconds. And so that gives you a bunch of time that you kind of bank to when you get to one of those, like for this picture, answer the next 6 questions. When you get to one of those, you can really stop and spend some time on it. All right. Consider the following molecule. Based on the elemental composition, which of the following molecules does this represent? It's got nitrogen in it, so it could either be a protein or a nucleic acid. It does not have phosphorus in it, so it won't be a nucleic acid. So this is an amino acid. Let's go find it. Uridine, a building. It's just weird. Glycine, building block of proteins. It's not telling you the word amino acid here, but it's just giving you the definition of amino acid a lot. AP does that a lot. This is the correct answer. And this is just a trivia question, right? What are amino acids made out of? In order to develop antibiotics, scientists need to understand how bacteria grow, divide, and spread during infection. An essential part of bacterial growth is RNA synthesis. I feel like we just had this question. Because RNA must be synthesized before proteins can be assembled. If RNA synthesis is blocked, bacteria will not be able to survive or reproduce. Makes me think of the word fitness. Which of the following questions will best direct an investigation on how antibiotics that inhibit RNA synthesis can be developed? And so what are we doing here? We know what RNA is made out of, right? Nitrogenous-based phosphate, ribose, uracil instead of thymine, single-stranded, right? So let's look. Chemical compounds block the 3' end of the growing RNA strand. Okay, this is a good one because we know that DNA and RNA is read 5 to 3, and the new bases are added to the 3' end. That's good. 3 to 5. Yeah. I think this is it. No, proteins are, no. That was a hard one. You do have to know this is the difference between the 5' and the 3' end, right? So that's important. The following excerpt describes the potential origin of the first living cells on Earth. And then there's a thing there. We have proposed that a primitive cell or protocell would consist of two components: a protocell membrane that defines a spatially localized compartment and an informational polymer that allows for the replication of inheritance. This is a wonky sentence. Of functional information. All right. Based on this excerpt, which the following questions would best direct an investigation on whether the informational polymer is DNA or RNA? Probably, does DNA and RNA have the ability to hold information or something along those lines? Let's go see. Which phosphate group is present in the structure? No. There's which 5-carbon sugar is present in the structure of them? DNA or RNA? Yeah, this is it. This is it right here. Purines, no. Functional, no. There it is. Because the difference is the sugar. Boop. Moving on. We got a picture here. We got two monomers, and their water's pulled out, right? Dehydration. And then they're joined together, synthesis, dehydration, synthesis, which the following best describes the role that water plays in the reaction here. Water is condensed out of the reaction. A dimer is created when a covalent bond forms between two monomers. Great. Bodies of water, such as ponds, freeze from top to bottom, forming a floating sheet of ice. This can help aquatic organisms survive during cold winter months. Which of the following statements explains why ice floats on the surface of the pond? Ice floats because it is less dense than water. And notice it's simple, right? You don't have to get in the weeds to know that. Now, you might have to get in the weeds to know why it's less dense. It has to do with, the polar forces pushing each other out. Positives and not liking the positives, and negatives not liking the negatives. And as the water molecules slow down, they kind of like freeze in place, right? And when they do that, they spread out. All right, some graphs here. This is what I like. Scientists grew chlorella, a single-cell photosynthetic algae, for 3 weeks in a lab culture. The culture medium contained dissolved carbon dioxide, nitrogen compounds, but it did not contain phosphates. All right. At the beginning of the 3-week period, the scientists measured the rate of synthesis of certain biological molecules. So if we don't have phosphates, we cannot make nucleic acids, right? And we see which of the following. So beginning and end. So we'd expect to see the end have no nucleic acids. Let's go find that one. B and C both have a lower end than the beginning, but since phosphates aren't found in proteins, here we are. Okay, so are there any questions? Unit 1 questions. We can go here. We can go to my my laboratory here, and we can look at anything that you guys want to look at in Unit 1. This is carbs, lipids, nucleic acids, proteins, macromolecules in general. Any questions about any of this content? We can talk about Avatar: The Last Airbender as being the best television show of all time. We can do that also. Probably not best use of our time since you guys have an exam soon. But, yeah, I think Unit 1 is pretty good as well. I think Unit 1 is pretty good as well. All right. I mean, there's not very many people in my stream right now, so if you guys have a unit you would like to see me cover, maybe that's a better way of doing it instead of going top to bottom. Unit 2 was one of my students' weaker units of the year. I don't know how you guys did, but Unit 2 was weak for us. So I'm fine to go straight to Unit 2 questions and start to kind of work through the content there. 5 and 6, yeah, 5 and 6 are usually a little lower for sure. Also, we can skip straight to those units if you want to. I am totally fine with that. 4 was hard. Okay, we can do 4. 4 is definitely one of those units that has some harder images. I think some of the graphics for 4 are tough. Pedigrees, okay, sweet. Yeah. Some of my students really struggle with pedigrees as well, because they're pretty much a logic problem. All right, let's do 4. We'll skip 2 and 3. I had a couple students that I knew were interested in some 3 questions, and so if they show up, we may go back and do 3, which is fine. I have no idea how many people are currently in here or anything, so all right, let's do that then. We'll go do 4. All right. Which of the following scenarios best describes a positive feedback mechanism? Okay, good. Feedback mechanisms are one of those things that you will see. So the way to think about feedback mechanisms is that a positive feedback mechanism creates a product, right? And that product stimulates the production of more product, right? And so think about it just continuing, right? It's always moving. It's always moving. My hand, you can't see my hand. Okay, it's always moving. As opposed to a negative feedback makes a product, and that product then goes back and turns off the process, right? And that's negative. So we're going to look ABCD here for something that turns or keeps something going, right? Several hours after a meal, blood glucose levels begin to drop, prompting the pancreas to secrete glucagon. Glucagon causes the liver to convert stored glycogen into glucose, raising blood glucose levels. And so notice what happened here. Blood sugar dropped, and so this happened to cause it to raise. And so it produced a product that turned off this. Off was low blood sugar, right? And so that is incorrect. After childbirth, high levels of prolactin induce lactation in the mother. The nursing before or during nursing, infant suckling triggers the release of more prolactin, which further stimulates. So this makes a product that's turning on the process, and you have this loop. Good. B. Correct answer. All right. And so when I say difficult graphics, here's a great example. My recommendation, more than likely, if you get a question like this, I mean, AP does weird stuff. So there was one year that I saw a multiple choice. It may have been on one of y'all's practice exams where there was an entire codon chart for one question. So whatever. But more than likely, if you get a graphic like this, you're going to have several questions associated with it. And so my recommendation is to spend some time, right? So think about it this way. Let's say that this says for the next 5 questions, use the graphic. And think about that 5 minutes is 7.5 minutes for you to answer all of those questions. That's a lot of time. And so spend a minute just familiarizing yourself with this picture so that you're not constantly having to go back up to it and like re-familiarize yourself with it, right? So let's look at it. Here you have a signaling molecule, epinephrine. Epinephrine binds with the receptor, right? Which is then going to activate. Anytime you see these arrows, it's just like activate adenylyl cyclase, which is going to in turn activate cyclic AMP, right? ATP is required for this. And cyclic AMP is then going to activate two processes, or one of two processes, depending on the situation. If it's active, it is going to make glycogen phosphorylase, which is going to cause glycogen to break into glucose. This is basically the idea of glucose being released into the cell or into the bloodstream, right? If it's inactive, then this enzyme here is going to it's also going to cause this particular enzyme, glycogen synthase, to stop making more glycogen, right? Because if you were making more glycogen, you would be countering what's going on over here. So cyclic AMP is turning on two things. It's turning on the process of glycogen being broken down into glucose and turning on this thing that's going to cause no more new glycogen to be made. And so now that we've done that, let's go look at the problem. If a toxin prevents the phosphorylation of glycogen synthase during this pathway here, right, which of the following will most likely occur when epinephrine comes into contact? So if you have a toxin that prevents this from happening, right, then what's going to happen? Then glycogen is going to continue to be made, right? Let's go find that. Glycogen synthesis will not be affected, right? So let's look. Glycogen phosphorylase will phosphorylate glycogen synthase to prevent new. No, that's not going to happen. The epinephrine receptor will no longer recognize. No, that's up here, so that's not going to happen. We like C. And again, D is way up here as well, so it's not going to go back on it. These pictures are why Unit 4 was a little more difficult, right? The content itself is the vocab and all that stuff is not very hard. Genetic mutation causes somatic cell to become cancerous. Which of the following is most likely explanation for how the mutation caused this malignant transformation? All right, so mutations, change in the DNA. Cancer is unregulated cell growth, right? And so what causes this? So we're just going to go see. Mutation created an underactive version of a protein that promotes cell division. This is the opposite of that. Mutation created an underactive version of a protein that promotes apoptosis. This could be it. Remember, apoptosis is the programmed cell death. And so apoptosis would be promoted in situations where cancer is present. And if you can keep apoptosis from happening, then that could be bad. Overactive preventing cell cycle, no. Overactive inhibiting cell division, no. B is the correct answer. The image below depicts a somatic cell nucleus before DNA replication. So we have a DNA replication before, so we have 4 chromosomes here. If the cell undergoes mitosis, which of the following correctly depicts the daughter nuclei that would result? So we just have 2 cells that look like this, right? I don't know why this is funny, but this is the correct answer. 2 daughter cells, each containing 4 identical chromosomes. Because mitosis just gives you 2 cells that are the same. So I'm just looking for this picture times 2. Yeah. So this looks like some kind of cascade. Yep, signaling cascade. So we have MEC phosphorylates to turn ERK on, phosphorylates that turns CMYC on, that causes the transcription of genes promoting cell growth and division. Okay. So MEC, ERK, CMYC, cell growth and division. After MEC is activated, it phosphorylates and activates ERK. ERK goes on to phosphorylate and activate a variety of target molecules, including transcription factors like CMYC that promote cell growth and division. Which of the following toxins would most likely inhibit the phosphorylation of CMYC? Literally anything back here, right? If you turn off MEC or if you turn off ERK, then CMYC is not going to work. A toxin that prevents the phosphorylation of ERK. This is correct. Let's look at the other ones. I know I answered that quickly. Though, if you know that A is correct, just mark it and go on. Don't read the wrong answers. They're just sitting there waiting for you to confuse yourself. Come on, please. Please read us so that you can overthink. Don't read them, but I'm going to read them now so I can explain why they're wrong. The toxin that prevents the removal of phosphate from ERK. No, we're adding a phosphate to ERK, right? Prevents the dephosphorylation activity of MEC. This is a phosphorylation activity. Toxin that prevents CMYC from activating ERK. That is not what's happening here, right? And so the other three answers are just bad. And so, yeah. Good. The following is an excerpt from a scientific paper written by Levi et al. Plant cells a barrier to virus exit and entry to the cell. Thus, successful infection requires that a plant virus encode movement proteins to transport the viral genome locally, cell to cell, within a leaf and into the vascular system, which the virus will systemically invade the plant, or systematically. So how do they get plant to plant, or cell to cell? Those are plasmodesmata. Let's go find that. There it is. I love like when you know the thing, and granted I've been teaching this content for a whole long time. But so let's go do this. Movement proteins increase the movement of transport vesicles between plant cells. Why can't vesicles form in plant cells like they do with animal cells? Because of the cell wall, right? The movement of vacuoles between plant cells, that's not happening. Vacuoles don't do that. Movement of proteins increase the surface area to volume of the plasma membranes. That's not a thing. This is not, because this also doesn't talk about cell wall. Permeability of plasmodesmata. Plasmodesmata are like little tunnels that connect to plant cells. I always talk about the interjoining closet that I have between me and the teacher behind me, who is the one and only famous Mr. Krug. We're very blessed at Murray High School. All right, the bacterium Vibrio cholera causes cholera, an infectious disease that often leads to severe dehydration as a result of ongoing diarrhea. During an infection, Vibrio cholera releases a protein complex called cholera toxin. It's a bacteria that releases a toxin. Okay, good. Yeah, Mr. Krug's, we've been teaching together for many years now. He's a dear friend of mine. He's a phenomenal, world-class teacher. And he might be streaming tonight too. So if you're an AP Chem, hop over to his stream. Tell him that I sent you. Do it. He'd love to hear from you. After its release, cholera toxin enters intestinal epithelial cells and binds to the G protein. This causes G protein to permanently be activated. Oh, interesting. The downstream effects of the G protein activation are shown in the pathway below. So let's look. So we have this toxin, right? This toxin binds here. There's the toxin, which activates this G protein, which is going to cause this, transduction here, which is going to cause chlorine to move from inside the cell to outside the cell. And what's going to happen then, right? This is a unit 2 thing. But if you have chlorine building up on the outside of the cell, you're going to have a hypertonic environment building up, and then water is going to leave the cell, and it's going to enter into the extracellular spaces, hence the diarrhea. Cause severe dehydration, right? And so there's that. Based on the information above, which of the following correctly describes cholera toxin and its effect on intestinal epithelial cells? Intracellular, it increases extracellular. I'm down with this. Extracellular chlorine ion. Let's see what else we got here. Is extracellular signal that decreases, because this is not an intracellular, right? Intra means within. So it's an extracellular signal that increases. Intracellular signal that decreases. I guess we're going with this. It's an intracellular signal. Sure, intracellular signal. Yeah. Okay. Some of the wording on this could use some help, but it's fine. All right, the endocrine system maintains homeostasis in the body through the release of various hormones. For example, the pancreas releases hormone insulin to help regulate blood glucose homeostasis. Which of the following best describes the role of insulin regulating blood glucose homeostasis? Interesting here, because our course description does not require that you know particular hormones, right? And so this is a holdover, I believe, from pre-2019 CED change or course description change. And so this is the kind of content that's not going to be required of you. An actual AP question would tell you what that's doing. But insulin, you could probably figure it out just because you know things, right? Insulin is released in response to high blood pressure, but it's a negative feedback loop. Not blood pressure, blood sugar. A partial hepectomy, surgical removal of a section of the liver, is often performed to treat tumors or other abnormal growths in the liver. Following the partial hepectomy, resting liver cells re-enter the cell cycle, duplicating until the excised portion of the liver has been regenerated. According to the information above, performing a partial hepectomy causes liver cells to exit G0 and enter G1. I like that. Nothing to do with meiosis, nothing to do with exiting mitosis. Good. And so basically, understand this, and so I can help you put this together with everything that we've learned. How do those cells know to be in G0 in the first place? Well, they're all talking to each other, right? That's a signal transduction response thing. And then when they realize, okay, the whole liver is done, then they all enter G0. And if there's damage to the liver and repair needed, then they get out of G0 and go into G1. And they're all talking to each other to coordinate this. Talking to each other. Oh, my lands. What is going on here? Look like somebody drew on the wall. To complete external fertilization in fish, sperm cells must swim through open water in order to fertilize the egg cells. During the process, females release a substance called ovarian fluid. Researchers hypothesize that ovarian fluid may play a critical role in attracting sperm cells toward egg cells. To test this hypothesis, researchers performed an experiment in which they observed the swimming paths of sperm cells as they moved toward egg cells. The researchers recorded swimming paths in water A and ovarian fluid B, A as shown. Okay. So A in water. This is in water, and this is in ovarian fluid. Okay. And so in water, they seem to be aimless, right? They're going in circles. I mean, if you go in a circle, you're just doing this. There's no purpose. Whereas if you're going in a direction, you're moving toward something. So it seems to suggest that this ovarian fluid is a signal that causes them to move in a certain way. Which of the following conclusions about ovarian fluid is best supported? It contains local regulators that help sperm swim straighter, making them less likely to fertilize eggs. It's not true. They're more likely to fertilize. Ovarian fluid contains long-distance signaling molecules that help sperm cells swim straighter, making less likely. No. Local regulators that help sperm cells swim straighter, making them more likely to fertilize eggs. We like this one. Let's see what else we got. Long distance. So good. This is kind of a weird question. Local regulators has to do with like local, like in an area, as opposed to one part of the body to another part of the body, something like that. So I like local, though I have no doubt that this one's going to be long distance, but this is just a bad question. The reason this is a bad question is because you aren't given parameters for these things. I like local just because of the way I'm envisioning this happening, but it's just a bad question. Yeah, they're gonna say long distance on this one. I probably missed this one when I went through last year too. Just not learn from my mistakes. All right. So any particular questions, Unit 4, that we can go back and look at? Anything that you feel like you'd like me to cover from Unit 4? You know, everything that we've talked about. Anything in the questions sort of trigger a question from you? Anything like that? I think Unit 4 is pretty straightforward. It is one of my, I think it's one of the better units of the year as far as our test averages were concerned. So, oddly enough, it's one of the worst units of the year with my honor students. So, are these notes available? Not like, I don't have like a paper, but I used these PowerPoints to make all of my videos. So I have videos on 4.1, 2, 3, 4, 5, 6 on YouTube. So you can go watch those, me doing the lecture. You know, they're about 8 to 10 minutes each. You can watch me on double time and I won't sound so slow and southern. So yeah. And I think with all the units I also have just a big Unit 4 review video too. If you want to just go and watch like just a video on Unit 4, you can do that as well. So, all right, let's move on to Unit 5, everyone's favorite unit. Yep, glad to do it. Love doing that sort of thing. It is a great joy of mine. I really do enjoy making this kind of content. It is a lot of fun. Okay, Unit 5, several of you guys mentioned Unit 5. Is there any particular section you want to look at for Unit 5? Or do we just want to, we could do both quizzes. We could do quiz 1 and quiz 2. Yeah, I mean, cramming, that's the way. That's why we're here, right? We're all cramming together. We're all just cramming together. Hey, if you have friends that you think, hey, my friend would really benefit from being here and watching this, you should bring them on in. Bring them on in. We can let's see, non-Mendelian. We could do some. Tell you what, let's do. Yeah, the chi-square thing. We can talk about chi-square for sure. I'll even, after we do Unit 5 stuff, I will go and look at a chi-square problem with you guys. So, all right, let's just do this Unit 1, or this quiz 1 here, since I think it's gonna cover pedigrees. We got 5 questions. Which of the following correctly describes how mitosis and meiosis are different? Mitosis is making 2 cells, right, that are identical, whereas meiosis or meiosis, however your teacher pronounces them, are going to make 4 genetically different haploid cells, right? So let's go find that. No, this is incorrect. It is not doubled after mitosis. DNA replication occurs prior. This is incorrect. Mitosis produces genetically identical. Meiosis produces genetically unique daughter cells. This is correct. Yep. Moving on. All right. Here's some data. The data above represents the results of 3 different crosses in which a single gene determines whether a certain organism is blue or yellow. So here we go. Which of the following best describes how this gene is inherited from parents to offspring? All right, so let's look at it. Here we have yellow male, yellow female. All of them are yellow, none of them are blue. Here we have blue and blue. 45 are blue, or 3 out of 4 are blue, and 1 out of 4 are yellow. And so what does this tell us? Right? If blue and blue are together, but you get yellow out of it, then that would mean that yellow is probably recessive, right? And that would make sense over here, because all of them are yellow, then you're only going to have yellow. And if you have a yellow male and blue female, 30-30, this would be like big A little a times little a little a. I do not believe this is a sex-linked trait at all, so we're going to say autosomal recessive here, or we're going to say blue is autosomal recessive allele because no blue offspring are produced in cross. No, yellow is autosomal recessive because of the 3-to-1 and cross 2. This is where we're going. Boop. So notice how you kind of have to like logic through that, right? You have to sort of take your time with this question. You know, some of those, like the question we just did, is like a 10-second question, right? And so spend a little more time with this one. If you spent 10 seconds on the last question, you have 170 seconds for this one, right? And so just park on it and like talk yourself through each of these situations. And you may think it's just 1 point, but it's not. It's 1 point, but it's also like a confidence building thing. Like you're building confidence because you know what you're talking about, right? And if you get to that question and you're like, oh crap, I don't know any of this, then just skip it. Just mark C or whatever and move on, right? Don't let that one question like derail your test. The following pedigree shows the inheritance pattern of a certain trait over generations. Okay. Let's see. So here, mom has it. Dad doesn't have it. Mom has it. Son doesn't have it. And so this means that it's not a sex-linked recessive trait, right? It's not sex-linked recessive because if this was sex-linked recessive, mom always gives it to sons, right? It is not a sex-linked dominant trait. I don't even know what we're doing now. I'm just talking through the pedigree since some of you guys had questions about them. So I'm just helping you understand where I get this from, right? We see both present in the children, right? And so it makes sense to me that this is a homozygous or a recessive trait, and autosomal recessive, and this individual is homozygous recessive, and this individual is heterozygous. Let's see if that plays out in the rest of our. It seems to play out in the rest of our pedigree as well. It's on a nuclear chromosome, not on a mitochondrial DNA. Yeah, Y-linked is only fathers passing to sons, right? And in all of my years teaching this class, I have never once seen a Y-linked problem. So this may be the year, but I haven't seen it yet. And so why is this a nuclear chromosome and not mitochondrial? If it was mitochondrial, then all of these children would have it, right? Because if it's mitochondrial, mom passes it to all of her offspring. So let's go find it. A female in generation 2 passed the trait to her female offspring. No. Male generation. No. Female generation 3. Female generation 3 pass the trait to the male offspring. Female generation 3 inherited the trait from her affected father. Okay, I'm reading this backwards. Is on a nuclear chromosome. So let's just, basically, for this one, we're going to look and see which of these is true sentence. A female in generation 2 inherited the trait, or gave the trait to her female offspring. She did, but she also gave it to her male offspring. A male in generation 3 inherited the trait from his affected mother. He did do that. But this doesn't prove that it's not nuclear. The female in generation 3 inherited the trait from her father. Boop, boop. There it does, right there. Because mitochondrial traits are not inherited from the father. There we go. All right, here we go. What is this? Numbers. And speaking of numbers, real quick, over the years, the need for a calculator has continued to decrease. And so I would say that, I'm not saying that you don't need a calculator, because you do, but your calculator is probably going to be used for like multiplication more than it's going to be used for like, water probability or water problems or potential problems, right? Just all of that formula sheet is almost completely unnecessary. So you'll probably use Hardy-Weinberg, and you'll probably use chi-squared. I hope that I'm wrong. I hope that you guys, all of the math that your teachers have quizzed you on over the semesters are going to be useful. But we'll see. A botanist is studying the genetics of a particular species of a plant. She knows that one plant may contain hundreds of individual seeds, each of which is the result of a separate fertilization event. She determines that the seed color is controlled by a single gene with 2 alleles. The dominant allele is black, the recessive is brown. In a follow-up experiment, the botanist crosses homozygous dominant plant with heterozygous. So homozygous dominant black with heterozygous black, we're going to get all black, right? Everything's going to be black. Which of the following is the best prediction for the frequency of brown? If we're going to get all black, then there's going to be 0% brown. Questions on that one. It worked fast. But I feel like it's big A times big A, little a on that one. And you get all big A. All right, good times. We got a fun one here. In fruit flies, Drosophila melanogaster, a mutation in a gene called ebony causes flies to have black body color. I thought they all did. The mutation for black body color is a recessive wild type for tan. I guess fruit flies are tan. Learned a thing today. And a different mutation called Actinopedia causes flies to have abnormal leg-like appendages on their foreheads in the place of antennae. The allele responsible for these leg-like appendages is recessive to the wild type dominant. And so we have tan is dominant and a regular antenna are dominant. All right. A wild type female that has the genotype this, right, which is heterozygous for both traits, is crossed with a mutant male that has this genotype. The expected and observed results are shown in the table. Offspring, genotype. Oh, so this looks like a chi-square, right? Yeah, expected 1/4 for everything. Observed is a lot more here and a lot less here. This is probably going to be due to linking. Let's go look. Which of the following best explains the difference between the expected and observed results of the cross? I'm going to say because E and A are linked together. And that's what I'm going to say. Are near each other on the same chromosome. Yep. All right. There's a formula for figuring that out. And if you take these two together, which are going to be the, these are the parental genotypes, and these are the non-parentals. And if the non-parentals are lower than 50%, then the genes are probably linked. And that's the way you need to think about it. It took me several years in my career to be able to explain that as well as I wanted to be able to explain that. I don't think this is going to be that difficult. Let's just go into the unit test then, and we'll move along. If you're just joining us, anytime you have a question, ask. We've just been going through con questions together, and I've been teaching content. But yeah, if you're just joining us, thanks for coming. Tomorrow evening, I'm going to be going through some FRQ questions. And so if you have requests, please ask. My current thoughts are I'm going to go through the 24 and 25 FRQs, right? Last year's exam administration and the year previous, and go through those FRQs with you guys and just walk through them, talk about strategies for FRQs as well. It's half the test, and frankly, I think it's where a lot of people can sink or swim. I think the FRQ is really important. So, all right, let's do this. Unit test, unit 5. Here's another fruit fly thing. There are multiple genes that influence eye color. One of the genes is located on the X chromosome. It affects the flies, has white or red. The allele for white eyes are recessive and to the allele to red. In addition, there are multiple genes of fruit flies that control wing shape. One of these is located on chromosome 2. Affects whether the fly has wild type or vestigial wings. Vestigial means that they used to be there, right? Or they're a vestige of the past. The allele for vestigial wings are recessive to wild type. True breeding female with white eyes. So true breeding female with white eyes is going to be 2 recessive alleles, right, on the X chromosome. And wild type wings. True breeding wild type wings, and so it's going to be big A, wild type wings, is crossed with a male that has red eyes. So the male has a dominant allele, but a single dominant allele, and also vestigial wings, which is going to be homozygous recessive. What proportion of the resulting offspring is going to be expected to be female with red eyes and wild type wings? Okay. So can we have a female with red eyes here? We can. Because the mother only gives away a little a, right, and the dad gives away a big A, all the female offsprings are going to have red eyes, in fact, because the females get their allele from mom and dad. Dad only has true breeding does mean homozygous, correct. True breeding technically means that it's only able to pass down one type of allele. Good. And so the females will all have red eyes. And so we're at 100% with our females, right? But we're at half because only half the offspring are going to be female. And so we're at 1 out of 2. It's going to be females with red eyes, right? With wild type wings. Well, all the offspring are going to have wild type wings because we have a homozygous wild type with a homozygous vestigial, right? And so we have 1 times 1/2, right? So we're using our probability math here. And so 1 times 1/2 is 1/2. Any questions on how I got that one? We don't do a lot of dihybrid crosses with sex-linked, and that's what this is. But notice what I did. I handled them separately, right? I dealt with my sex-linked thing and got a probability over here. And then I handled my wing thing and got a probability over here. And those two probabilities are exclusive, and so you multiply them together, right? And so 50% of the offspring are going to be females with red eyes, and 100% are going to be are going to have wild type wings. And so I go one half times one. This is the kind of question that will slow you down for sure. Some of you guys can probably get this quicker than I could, and I went through it pretty slow, but yeah, some of you could solve it quicker than me. Others of you, this is going to really bog you down. And so, just again, don't get bogged down on, don't look up and you've spent 5 minutes on this one point. All right. The diagram shown below shows a non-disjunction event. Non-disjunction is a failure of chromosomes to separate for a single human chromosome during gamete formation. The chromosome depicted diagram gene A. Okay. If gamete 2 is fertilized, how will the non-disjunction event affect the resulting embryo? The resulting embryo is going to have 3 chromosomes instead of 2, right? 4 copies, no, non-viable because of monosomy. Not monosomy, it's trisomy. The embryo will have a higher than normal of gene A, right? That's correct. Gene A is going to be double in that particular, and will not be able to produce gene A. No, C. Good. Okay. Which of the following statements describes an example of phenotypic plasticity? Phenotypic plasticity has to do with a variation of phenotypes across of environmental conditions. Pretty sure that's correct. Yeah, that's correct. Well, you know what? Unit 5's been a little while. Lizard has green skin when raised at 20, and also, so this would not be correct, right? Because this is a changing environment, but non-changing phenotype. Semi-aquatic plant produces large, thin, submerged leaves and small, thick. No. Oh. This could be it, because the submerged leaves versus aerial leaves. We like this one. Sheep has long wool before being sheared and short leathers. Yeah, it's got a haircut. Person develops a gene mutation in a healthy lung cell result. No, so this is correct, right? Because you have the change. The change in environment represents a change in phenotype. Cystic fibrosis is an autosomal recessive disease caused by mutations in the cystic fibrosis individuals with cystic fibrosis have a sticky mucus in their lungs and other organs. The mucus can lead to infections, additional complications. And so there we have our key to our pedigree. Which of the following pedigree shows a likely pattern of inheritance in a family of cystic fibrosis? Okay. So if it's autosomal recessive, that means you're going to only inherit it if you get 2 recessive alleles. This one's not possible, right? Because you have 2 infected individuals. They would both be homozygous recessive, but 2 of their children don't have that. Same here. You have the infected individuals, but non-infected or affected parent or children. And this one can't happen because of the same thing here. Here we have a good pattern of inheritance because, and notice A, B, and C all were eliminated because of the same thing. Right, and so D is correct, and we don't even have to know why D is correct. These are obviously wrong, but let's look and see why D is correct. Can you have these 3 offspring from these 2 parents? Yes, you can. You have a heterozygous individual here or homozygous dominant, you don't know, but it's probably heterozygous because this child has a child that has it, right? And so you have a 2 heterozygous and a homozygous recessive. All the children are normal, but they're all heterozygous for the trait. And then this individual's heterozygous. This man is also heterozygous, and they have a child that has it. Same situation over here. D is going to be correct. Yeah, pedigrees just take a lot of times. You have to think about working backwards with pedigrees. Gametogenesis, or the formation of genes, occurs when diploid germ cells undergo meiosis. In fruit flies, Drosophila melanogaster. Hopefully you pronounce that while you're reading it. Diploid germ cells contain 8 chromosomes. Which of the following correctly describes the number of chromosomes the daughter cells produced during gametogenesis? Describes the number of daughter cells produced. Since this one contains 8 chromosomes, 2n equals 8, then n equals 4. This is going to be wonky, I bet. Let's go and look. Daughter cells each contain 8 chromosomes after meiosis 1 and 8. No. Daughter cells contain 8 chromosomes after meiosis 1 and 4 after. This is good. 4 after and 2. No. This is good because, remember, after meiosis 1, you start with 8, but because of interphase, you end up with 16, right? Because DNA replicates. And so after the first division, you have 8 again, and then 4. So this is correct, but let's look at these. Contains 4. No, it doesn't. What did I do? Did I do the wrong one? I don't like this question. Fruit flies, diploid equals, so 2n equals 8. After meiosis 1, each one's going to have 8. And they're going to have 4 after meiosis 2. I don't know why this is wrong. Daughter cells contain 8 chromosomes after meiosis 1, 4 after meiosis 2. Let's just get it. Let's just pick them. The number of chromosomes in the cell is halved during meiosis 1, maintained during meiosis 2. I just don't think that's correct. So it's a bad question. Fragile X syndrome is a genetic condition that may cause delays in the areas of learning, language, and social skills. Fragile X syndrome is caused by a dominant allele of the FMR1 gene and is located on the X chromosomes. Individuals who inherit a copy of the allele typically display the signs of the condition. It does not tell us if it is. Does tell us it's dominant allele. So it is sex-linked dominant, which shows the likely pattern of inheritance. So the mom doesn't have it, so she's homozygous recessive. Neither of her children have it. Dad does have it, so it is dominant. Gives it to his daughters for sure. The daughters have it, but in this case, they only have one. In this case, she only has one copy of the allele, so it's possible that she'd have sons that have it and some that don't have it, which is correct. And is it possible that she would have daughters? And yes, this is good. This is good. With these pedigree problems, you do want to be careful because you can get bogged down in them. But we can go down here and eliminate these as well. Everything's normal here. Both parents have it. All their children should have it because they're all getting the dominant from dad, or at least the daughters are. Both daughters should have it here. It is possible that the sons could split here. Same thing over here. Dad has it. Daughter should have it. D, let's see why D is wrong here. There's no way that this son can have it. So that's why D is wrong, because mom is homozygous recessive. There's no way the son can get it. A is the correct answer. This one's a little slower. Hopefully you guys are sticking with me. The group of researchers are studying stem cell inheritance in the garden pea plant. They obtain a tall pea plant of unknown ancestry, allowed to self-pollinate and collect 80 seeds. After planting the seeds, they observed growth of 61 tall plants and 19 dwarf plants. All right. So they allowed to self-pollinate, which means that, kind of self-fertilized. And so it was tall, but it was able to make short, then it's heterozygous. Stem length is inherited, incomplete dominance. Stem length, the dwarf is a recessive trait. This is good. There's a snowshoe hare. Displays a seasonal change in coat color from brown to white. Transition occurs as a result of changes in gene expression during autumn, which is when the hare's summer coat is replaced with winter ones. The timing of this transition allows hair to remain camouflaged. A group of scientists studied the certain snowshoe hare population in western Montana for a period of 3 years. And there you have the percent of the white coat and the date, and then, there it is. Okay. We got some stuff. Based on the information above, which of the following statements is most likely true about coat color transition in snowshoe hares? So phenotypic plasticity, right? Pleiotropy. Transition from brown to white occurs as the genes for having a white coat become more common in the population. No. The transition from brown to white is an adaptation to climate change, which is predicted to nope. The transition from brown to white result of gene expression changes induced by autumn day lengths. Yeah, the prompt essentially tells us that, right? It doesn't add the idea of day length, but it's the only thing that it adds. It's the only thing that this adds. So it's due to gene expression changes, but influenced by the amount of snow on the ground. No. All right. Diagram below shows two pairs of homologous chromosomes in the diploid cell. Larger chromosomes have different alleles of gene A, and smaller chromosomes have different alleles of gene B. So there we go, gene A and gene B. If the cell undergoes meiosis, which of the following is true regarding the allele combinations of the resulting gametes? So you could have big A, big B. You could have big A, little b, you could have little a, big B, and you could have little a, little b. 4 different combinations here. The allele combination big A, little a is just as likely to occur. No, it's not going to occur. Little a, little b is less likely. No. Big A, big B is just as likely as this one. This is correct. They're all 1/4% chance. During meiosis 1, pairs of homologous chromosomes align randomly along the metaphasal plate before being separated. This process helps generate genetic variation in offspring of sexual reproduction. How do I know which is more or less likely? Okay, good. So we'll go here real quick so I can write something. And so in that particular question, I'm not used to this one, so you'll have to excuse me. There we go. In that particular question, we had this situation, right, where this was the genotype of that individual, correct? And this is a 2n equals 4 situation, right, where this individual has 4 chromosomes. This is one homologous pair, and this is the other homologous pair. And we know that during meiosis, one of this pair is going to join with one of this pair, right? There is a possibility that this would happen. right? Because 1/2 and 1/2 could get together. But there's also a possibility this could happen. There's no possibility this is going to happen because the only possibility of this happening is if non-disjunction occurs, right? There is a possibility that this would happen, this little a and this big B. And there's also a possibility of this happening. What is the chance that this is going to happen? Well, big A is half of this and big B is half of this, and so that's a quarter. And you could do that for all of them. And so all of these are 25% likely to happen. And so they're all equally going to happen. That's the idea. I mean, this is like how you would, if you're doing a Punnett square like manually, which I don't teach my students manual Punnett squares at all, but all teachers do it different. I'm not saying I'm right or wrong. But if you're setting up one manually, this is how you would do that. I teach my honor students that, but not my AP students. All right. All right, good question. Really good question. During meiosis 1, homologous pairs align. Oh, we read this already. Which of the following explains how random alignment of homologous chromosomes during meiosis helps generate varied offspring? And so this has to do with what we were talking about, the separation of homologous pairs, right? It's in meiosis 1. Random alignment occurs as likelihood of crossing over. It's not between non-sister chromatids. This is crossing over doesn't happen. It happens in prophase though, so increase the number of possible chromosome combinations in the gametes. I like B. Formation of some diploid and some haploid, no. Different numbers of chromosomes. B is the correct answer. Good. All right, we missed one question. That was a bad question. At least I couldn't see why it was wrong. Why I was wrong, but it could be wrong. Okay, questions. Other questions on Unit 5. Any of you guys have anything to bring out on Unit 5 that we did not discuss? Seeing none, we'll move on to Unit 6. We're going to move. I know we don't have a lot of time, but following diagram. So this is Unit 6 questions on genetics, genetic expression regulation. Yes, I can go over haploid and diploid. Let's do that. Don't know why I did that. Okay. Okay, so here's our haploid diploid idea. A diploid cell, right, has both copies of chromosomes, 2 copies of each chromosome. Where did these come from? Okay, that is what you need to ask yourself. Like, why does it have 2 and not 1? The reason that this one has 2 is because one of them came from mom and the other one came from dad. Okay, and yeah, we'll look at after S phase 2. Good. All right, and the same here. This one came from mom, this one came from dad. A haploid cell exists after homologous pairs separate, which occurs in meiosis 1. And so haploid cells only exist, as far as we're concerned in this class anyway, right, as a result of meiosis. There are some exceptions to that out there in the world, but not really. Meiosis is what makes haploid cells, though some organisms live as haploid organisms. A diploid organism is going to produce haploid cells only as a function of sexual reproduction, right? And so after S phase, Here we go, right here. And so this is our cell, right? This is our cell. This is a diploid cell. 2n equals 4. Where'd I get 4 from? 1, 2, 3, 4. Right? One of these came from mom. One of these came from dad. Mom, dad. After S phase, each one of those chromosomes is going to duplicate. Right? This one duplicated. Now we have sister chromatids here, right? These two have, this one has 2 pieces. They're identical to one another. This one over here has 2 pieces. They're identical to one another. These 2 chromosomes, this one here, let me mark them with a red mark. Okay, these 2 chromosomes with a red mark are not identical chromosomes. They're homologous. They're not identical because one of them came from mom, the other one came from dad. Mom and dad are not identical to one another. And it's the same thing as these 2, right? These 2 are the blue chromosomes. I marked blue. Are not identical. They're homologous pairs. But this one now has 2 pieces that are identical. These are sister chromatids. These 2 are identical. And so during meiosis 1, first of all, crossing over happens, right, during prophase. But notice what happens. You have a separation. And this is why I struggle with that one question, because, like, look, after meiosis 1, you still have 4 here. It was, okay, I know why I was wrong now, but never, I'm just gonna get over it. I was counting these individual chromatids. Okay, but we're good. It should, okay, whatever. All right. And so these homologous pairs separate, right? And so this individual has 1 red and 1 blue. So does this one. And then after meiosis 2, that is when you have your actual haploids that have the individual pieces, right? Questions there? Was that a good explanation of that? Hopefully so. All right then. Yep, and haplodiploid's a pretty it's a pretty important concept, especially for Unit 5, but really moving on through 6 and even 7. So anytime you're dealing with the genome, that's an important concept. All right, then let's look at Unit 6. My students generally did pretty good on 6, so we'll look at it. Following diagram shows nucleotide base pairing in a double-stranded DNA molecule. The nitrogenous bases of the 2 nucleotides are labeled. Okay. Purine, pyrimidine. Nitrogenous base is a pyrimidine. Oh, man, I'm gonna get those backwards, I bet. I think purines are two rings, and pyrimidines are 1 ring. Yes, that is correct. And so this is a pyrimidine, this is a purine. Let's go find that. Nitrogenous base 1 is a pyrimidine, nitrogenous base 2 is a purine. I don't know how to remember that, honestly. There's not a good way to remember that other than just to know it. Though somebody in the chat may be like, this is the way my teacher says that we remember it, and you know what? That might be helpful. But obviously I still need help with it because that's just how unimportant it is. A scientist constructs a recombinant bacterial plasmid that contains a gene conferring resistance to the antibiotic tetracycline. The scientist transforms cultures of E. coli bacteria with recombinant plasmid, then incubates the cultures overnight on plates that contain media with and without tetracycline. A longer word, it's a shorter structure. There you go. That's a great way to remember that. That's good. Long word, short structure. I'll probably remember that myself. All right. Assuming the transformation protocol was successful, which of the following best predicts the bacterial growth pattern expected after the incubation period? Okay, so we would expect we would expect bacteria to grow on both, right? Because they're going to have the plasmid that's going to give them resistance to tetracycline, and so it should be able to grow on one with and without tetracycline. So we should have high growth on both, is my prediction. We don't have that option. Let's see. Oh, it's going to be some bacterial growth because not all the bacteria are going to take up not all of them are going to take up the plasmid. Right, okay, good. And so the one without, you should have high, and the one with, you should have some. This is where we're at. Okay. And so, again, just my reasoning on this one. The reason it's some here is because not all of the bacteria would take up the plasmid and be transformed, as we demonstrated in my own class this year when we only had like one little thing transform. I am the worst lab technician in the world. VKORC1 is an enzyme that promotes blood clotting, is encoded for that gene. I'm not going to say it more than once. And this is having this thing that you can't pronounce is on purpose, by the way. It makes it harder. And so I would just recommend you not pronouncing it. Just sort of like look at it and recognize it, right? Or come up with a different name for it. RC1, that's better, is an enzyme that is encoded by the RC1 gene. That makes sense, right? In some situations, doctors may want to reduce patients' RC1 activity because it promotes blood clotting, right? Activity to prevent dangerous blood clots from forming. To do this, doctors can use a drug called warfarin that inhibits RC1 activity. However, determining the correct warfarin dose can be difficult because individuals vary in their sensitivity to the drug. One cause of variation is genetic. Some people have a mutation in the RC1 gene promoter that decreases the level of RC1 gene expression. Doctors have found that in order to reach desired low level activity, patients who are homozygous for this mutation require less warfarin than those who are heterozygous. So that means it's probably a recessive gene. Okay. No. Because if you have this gene, it causes blood clotting. And if you're homozygous, it requires less of that. It's because there's less of this happening. So it is a dominant. It's on the dominant allele. Okay, which of the following best explains why homozygous individuals require a smaller dose? My guess is because the dominant allele is, this is on Unit 5 though, so we need to think about it differently. Just dosage. Let's just go look and see what our choices are. Homozygous individuals are less VRC protein in their cells for war. This is correct, right? So I was trying to overthink it, and it was just didn't need to do that. Don't do like me. Don't overthink it. The following model represents the elongation stage of the protein translation. Elongation is just adding. Elongation is adding amino acids to the ever-growing chain of amino acids. Yeah, so you said it's straight Unit 5. It presented like a Unit 5 question, right? Because of the homozygous heterozygous, but in fact, you didn't even need that. I wanted to make it more complicated than it was, but in reality, homozygous just had less than heterozygous, and you don't need to know why. The question didn't, I was trying to figure out why that's the case, and you didn't need to know why. So it's a great example of just overthinking, and you shouldn't do that. I do it too sometimes. Yeah. It's really easy to do, right? It's really easy to do. Which area represents the tRNA leaving the ribosome after transferring an amino acid? 4? Is leaving? No. This is weird. This is in sequential order. Okay, it's coming down, adding the thing. Here it goes. It's leaving. This one is coming down to bring purple. This one just brought red, and now it's leaving, so 3. What a weird question. What a weird question. All right, the following diagram presents the structure of the human L2 gene. Just again, in the diagram, rectangles represent exons and introns, or exons, and the lines represent introns. We'll talk about that in a second. Mutations in the sequence of L2 gene have been associated with certain types of lung cancer. The group of scientists proposes that another cause of these cancers is alternative splicing of pre-mRNAs. To test their claim, the scientists prepare a sequence of mature L2 mRNAs in lung tissue and cancerous tissues. Which of the following pieces of experimental evidence will be best support the claim that alternative splicing in L2 pre-mRNA is associated with lung cancer? Okay. So alternative splicing is the idea that the exons are put in different order, right? As opposed to 1, 2, 3, 4, 5, you'd have 1, 3, 2, 4, 5, or something like that. Because when you take the introns out, you're mixing up the exons. And so, which pieces of evidence will support the claim that splicing is a cause in lung cancer, that we would find a lot of different order in lung cancer cells, right? Let's go see that. Are without exon 3 both present? With and without exon 3 are, no, that's not. With intron 4 are more common in tumors? No. Because we're wanting to find the tumor one, we're wanting to find something different than 1, 2, 3, 4, 5, right? Or more without exon 2 are more common in tumors. So there it is. Okay. Good. Here we have a lac operon. So there's a promoter. We have some genes over here. We've got a bunch of business in here. Okay. And this tells us what all this stuff is doing. Lac operon is a coordinated or coordinately controlled set of genes. What a fun group of words. Required for lactose metabolism. The operon is associated with regulatory sequences are shown in the diagram. And this tells us what everything's doing. The activity of the lac operon is influenced by the availability of lactose and glucose in the intercellular environment. In the presence of lactose, the lac repressor protein is released from the operator, which allows the structural genes to be transcribed at a low level. If glucose, the preferred carbon source, is absent, the catabolite activator protein binds to the cap site, causing the structural genes to be transcribed at a higher rate. So basically, if there's no glucose, it's like, okay, we need to break down more lactose, right? That's the idea. Which of the following is the most likely result of mutation in the lac operator that prevents the release of the repressor gene? Okay, so if the repressor gene stays down on this operator, then none of these genes are going to be transcribed, right? And so you're not going to have beta-galactosidase, lactopermease, or galactoside trans that protein. You're not going to have any of those proteins. No, it won't. It won't be produced. Between hydrogen covalent bonds? Yes, I can do that right now before we answer this question. So a hydrogen bond is an interaction between two polar molecules. Notice I said the word interaction, right? Two polar molecules like water, is a great example, are interacting with one another. They're not exchanging electrons. They're not physically connecting to one another. They're just interacting. whereas a covalent bond is a physical connection between two structures. The way that I explain these bonds in my own classroom is I will use my whiteboard as an example. I have a whiteboard that has erasers on it, like the magnet kind. And the whiteboard, I said the whiteboard is connected to the wall using screws, right? There's a physical connection between the whiteboard and the wall. That's like a covalent bond. Covalent bond is sharing electrons. The oxygen and the hydrogen in water share electrons, right? And so they're covalently bonded. Whereas the eraser sticking to the whiteboard is not a physical connection, right? You can take the eraser off the whiteboard really easily, but there is an interaction between the eraser and the whiteboard. That is more like a covalent bond. It's not sharing anything. It's just more like positives and negatives being attracted to one another. It's a quick and dirty definition. All right. So again, we're looking for these proteins not to be produced. That's why A is wrong. Cabalate activator protein will be bound to the cap site even in the presence of glucose. In the presence of glucose, it is active. Right. No. Expression of lac L gene will be repressed even in the absence of glucose. That is correct. Well, no, lac L, no, incorrect. The transcription of structural genes we blocked even in the absence of glucose. This is correct right here because these are our structural genes. D is the correct answer. Okay, good. Remember, this is a reading test. There's some content that's required, but this is just literally can you read. You don't really have to know anything about like operon to get this correct. Just be able to read and look at a picture. In DNA replication, which of the following events happens during both leading and lagging strand synthesis? Right, so we're going to have some things that are common to both of them. Separates complementary strands, ligase, no Akasaki. This is helicase, so I think this is wrong. Synthesizes single continuous, no RNA polymers help to send. This is correct. Because this is just wrong, right? It's helicase. This happens only on the lagging strand. This happens only on the leading strand. And so D is the correct answer. Even if we don't read it, it's still correct. But we read it. Following diagram shows a double-stranded DNA molecule. There it is. Which of the following represents the correct mRNA molecule that will result from these stupid words? Okay. The coding strand is going to be, the non-coding strand is what's going to serve as the template here. All right, so we're going to see an A, U, G, C. I mean, these three didn't even start with A, G. This is a vocab question. I don't like these terms. The reason this one's called the coding strand, because it's similar to the code, but it just doesn't have any T's in it, right? So the coding strand and the mRNA should be identical except for the T's and the U's. I got it wrong. Oh, it's because I got it. It's 3 to 5. Yeah, this is correct other than the 5 to 3 thing. So stupid. Because this is 5 up here, then this one's going to be 3. Right, so it's just literally the same thing backwards. Okay. So that's I didn't mix up the everything I said was correct. It was just the 3 to 5, 5 to 3 thing. Remember that DNA is, and these are anti-parallel, right? They go in opposite directions. Yeah. Okay. All right. Woolly mammoths are extinct relative of modern-day elephants. Scientists have uncovered a number of woolly mammoth specimens preserved in Arctic permafrost and extracted DNA from their hair shafts of all these animals. This DNA has been studied in a laboratory using the same techniques used to study DNA of modern organisms. For example, a group of scientists used woolly mammoth DNA to sequence the animal's nuclear genome. First, the scientists cut the sample of DNA into fragments. Then the scientists amplified the fragments for sequencing. After sequencing, the scientists used computers to reassemble the fragment sequences to determine the order of nucleotides. Which of the following techniques have been accomplished? The amplification step. Remember we talked about this is a biotech was a 6.8 question here, and amplification is taking a small thing and making it into lots of things, and this would be PCR. Hey, here's a whole codon chart for one question. Here's our nucleotide. Using the following codon, translate the mRNA molecule into the corresponding polypeptide sequence. Okay, so this is our messenger RNA, right? And so we're going to have a codon here. I'm waiting for it to trick me. 5' cap is what connects to the ribosome. And so this is going to be read first, right? However, notice here that our start codon is here, right? So that's where we're going to start. And so this is going to be met. This is the trick, I think. U, which would be phenolphthalein. So let's go down here. Met phe. I'm not going to do the rest. Right. So the trick was, again, not starting here, but finding the start codon and then moving from there. Okay. Sweet. Questions Unit 6. Anything that's come up? You guys have any questions about it at all? Anything at all? We'll move on to Unit 7 if there's nothing. Again, tomorrow, if you're just joining us, we're going to work through some FRQs, and hopefully you will find that helpful. Working through last year's exam FRQ questions, 2024. Look at those as well, because they deserve a second look and a third look even. And if you're looking, if you want to just, if you're not going to be here tomorrow night and you want to do that, I have FRQ videos where I've solved those FRQs. This is Unit 7. Let's go do some. Yes, Unit 7 is pretty easy, except for some of the vocab can be a little weird, and of course, if you come across a Hardy-Weinberg problem as well, that can also be a thing. So that would be the only, Hardy-Weinberg's kind of a skill-based thing, but most of them aren't that hard, though. And a lot of teachers don't even teach Unit 8, which is unfortunate because Unit 8's a lot of the exam. So my Unit 8 review video is like one of my most popular videos, and I think it's because a lot of teachers don't teach it. All right, so let's look at this. As a result of adaptive radiation, there are roughly 150 species of anolis lizards found in the Caribbean Sea. These species differ in their physical features, behavior, and habitat use. Which of the following statements best explains how adaptive radiation could have led to the diversity? So adaptive radiation is the idea that you start with one species and it radiates out because of changing environment and or available niches. And so we're going to see if we can find that. Over time, many closely related species migrated to the islands from the mainland. Each species settled into an ecological niche that was best adapted for. I like that one. And I probably should have read the other one. Members of a single species floated to the islands from the mainland. Over time, the species diverged into many species or groups adapted for different. Literally, this one's just better. It's kind of what I said, without being too hasty. Yep, I get hasty. So I think the problem with A is the many closely related lizard species. This is the problem. This is just wrong. It's the idea of a single thing, which is what I said in my explanation, going out and radiating out to many things. You know, we got to keep the teacher humble. Keep him humble. All right, which of the following describes the result of artificial selection? Remember, artificial selection is where humans choose the traits that they like based on their own preferences. And so let's go find that. Frequency of gray-feathered tawny owls is decreased due to climate change. No. Ear length in a population of dogs increased due to selective breeding. B is the correct answer. Moving on. Weeds growing among crop plants can cause significant losses in... When do we put a line of best fit on the graph? That's a good question. I have not seen a question that asks you to draw a line of best fit. That is beyond the scope of this class. However, the only time that you would see that is if you were dealing with like a scatter plot. And so, let me see here. I'll show you a scatter plot real quick. You're not going to have to draw a line of best fit. But the only time you would see something like that is if you're dealing with a scatter plot. And what is a scatter plot? Well, I have it here. It's a correlation between variables, seemingly unrelated variables. So here we have the sale price of apartments versus the brown living area, right? Nothing to do with biology. But these two variables are seemingly unrelated, right? And what you do is you plot a bunch of dots and you see, well, there seems to be a correlation here, right? And you could draw a line of fit here. If you were in a stats class, you could draw a best line of fit. This line of fit would get like a D minus for being terrible. So would my D minus also get a D minus. But in this class, you're never asked to draw that line of fit ever. So, good question. Good question. Hopefully you weren't asked to draw a line of fit in your class this year. But you know what? I have discovered that many teachers do things many different ways. That's what I have discovered. Good question. All right, weeds growing. I've already read this. No, I haven't. Weeds growing among crop plants can cause significant loss in crop yields. One way that crop farmers can remove weeds is through the use of herbicides. One class of herbicides, acetolactate synthase, ALS inhibitors, target enzyme ALS. Normally, the ALS enzyme carries out the first step of valine, leucine, and isoleucine synthesis. These are amino acids. However, when a plant's ALS enzymes are inhibited, the plant cannot make these amino acids and it dies. Which of the following best predicts what will happen to a weed population in a field that is over time consistently sprayed with ALS inhibiting herbicide? So we would predict that a mutation would increase the fitness, that if there was a mutation that caused those plants to make those amino acids anyway, that particular strain of plant would be increased in fitness. So let's go find that. The weed population will evolve resistance to the herbicide, likely through the selection of plants with mutation of the ALS gene. It's just literally what I described. It's probably wrong though, because I read it fast. Nope, it's not. Okay, good. A group of scientists is studying a certain gene in a population of fish. This gene has two alleles, dominant Y, recessive little y. The scientists collected allele frequency data over a 12-year period. The results are shown. So you have a table in which the dominant allele is present 43% and the recessive allele is 57%. And then in 2018, 12 years later, you have a shift toward the recessive allele. All right, and we know that the definition of evolution is a change in allele frequencies over time. So this is evolution. probably because the recessive allele, presence of recessive allele, increased fitness in those individuals. Let's go find out. After analyzing the data, determined because the allele frequency changed, the fish population was not in equilibrium. Right, good. Which of the following is the most likely reason that a population deviated from Hardy-Weinberg expectations in the study period? Because something was different, right? Those are 5 things for Hardy-Weinberg. Let's go see. Fish environment was stable. That's not true. There was no migration, not true. It was consistently large, not true. Female fish in the population preferred mates. This would not be random mating, right? This would be against Hardy-Weinberg. D is what we're looking for here. Which of the following statements supports the hypothesis that dolphins are more closely related to humans than fish? That means they're going to have more in common with them structurally. We're going to see dolphins and fish have fins for swimming in aquatic environments. Humans have legs. That would be convergent evolution, or analogous structures does not show common ancestry. Dolphins and humans have gills. No, we don't. Dolphins and humans have 4 limbs with similar bone structure, whereas fish do not. That's homologous structures, does denote common ancestry. What is this one, though? Dolphins and fish have more nucleotide differences in their ribosomal RNA than dolphins and humans do. Note there are 2 answers to this question. Okay. I was about to say. There's not going to be a question like this, by the way. Choose 2 answers. I didn't even read this part. You're not going to ever have a choose 2. But I was like, okay, both of these can't be wrong because they're both right. The following cladogram shows evolutionary relationships among 6 birds. Good. According to this cladogram, malleefowl are more closely related to partridges than to magpie geese. So here's magpie or malleefowl. They share a recent common ancestry with partridges, where they do not share a recent common ancestry with magpie geese. Right? You can see that here. They're way down here is their common ancestor. Which relationship best justifies this? More closely related to the outgroup than nope. Are more malleefowl share more recent common ancestor with partridges than with magpie geese. That's what I said. Here's a line of fit right here. There it is. And this is a scatter plot. Reduction in speed, time since first exposure. There's a negative correlation between these 2 things because it's going down, right? So there's a line of fit. Cane toads, Rhinella marina, have a toxin in their skin that can kill their predators. These toads were introduced to Australia in 1935. Since then, Australian populations of cane toads have grown considerably, with the toads now considered invasive. Australian red-bellied black snakes, that word, eat small animals, including toads. These snakes have not been exposed to cane toads prior to 1935. Researchers predicted that the introduction of cane toads would have placed a selective pressure on Australian snake populations that would favor the with inheritable resistance to cane toad toxin. Sure. To test their prediction, researchers determined the levels of toxin resistance in several populations of red-bellied snakes and have been exposed to cane toads for the amount of time. Resistance levels were approximated by measuring how much snake swimming speed was reduced after being exposed. Okay. Smaller reduction in swimming speed meant higher resistance. Okay. So over time, we have a decreased reduction in swimming speed, which means that we have an increased resistance over time. Okay, good. Based on the experimental results, which of the following statements about red-bellied black snake evolution is true? In areas with cane toads, snakes with higher toxin resistance produce more offspring than snakes with lower resistance. Yes, because a higher toxin resistance is going to increase fitness in this area. Good. RNA world hypothesis proposes that RNA is the earliest self-replicating biological molecule performing functions in early cells that are now fulfilled by biological molecules. One piece of evidence supporting this is that RNA molecules can encode genetic information. This provides support for the idea that in early cells RNA carried out function similar to that of DNA. Which of the following is an additional piece of evidence in support of the RNA world hypothesis? Like proteins, RNA can carry out catalytic activities. That's right. The idea of ribozymes, right? They cannot capture light. They cannot physical support for cell walls, and they cannot make the structure of a membrane. Good. Ribozymes, there it is. Which of the following does not describe a human activity that is likely to contribute to the extinction of the coral reef? Okay, so we're looking for something that would not kill a coral reef. A scuba diving instructor doesn't educate. That would kill. Government establishes a marine protected area that restricts the type of activities. This would help, right? We're looking for something that would help. Good. And that question back there, I wish I could go back. It's a perfect example of like just tricky wording, a simple concept, right? The idea that the government protecting a place to help coral reefs, it's a simple concept. The reason you're going to miss that question is not because you don't understand that concept. It's because the wording is tricky. This is a reading test first, right? Because if you cannot read the question carefully, it doesn't matter if you know the content. And so just be careful. And the reason that I've missed a lot of the questions that I have missed tonight is because I'm not carefully reading. I'm demonstrating the thing that I'm preaching against, right? And so make sure that you read carefully. That question was 5th grade content, but high school level comprehension level, right? Which of the following is an example of gene flow? Gene flow is the movement of genes from one population to the next. Let's go find that. Brown owls in a population have more offspring over a multi-year period than gray owls. No. Hurricane reduces the size. That's genetic drift. Genetic frequencies in a population of elephants fluctuate over time. Nope. Due to chance. This may be. Let's go see if D is better. Sea anemone floats from an area inhabited by one population to another population. Okay, good. So gene flow is the movement of genes from one population to another. It's just vocab question. Destruction of swampland has reduced the area inhabited by certain panther populations. As a result, panther population is small, highly inbred, and risk of extinction. This is an inbred population, right? It's a genetic drift because of habitat reduction. Which of the following actions can humans take to decrease the panther population's extinction? So again, here we're looking for things that we could do to that are positive, right? Add a species of large cat. Nope. Introduce a small number of panthers from a genetically distinct population to the at-risk population. This could be good because it would increase genetic diversity. Let's go look. Use the fittest male and female from the population in a captive breeding program. That will no, because this is going to increase the bottleneck, right? Move half the population to a different isolated swampland. There's a little no, because this is also going to cause a bottleneck as well. Good. Okay. You know what? I've got time for Unit 8. Any particular questions on Unit 7? I don't think so. Unit 8 and 7 and 8 are not too bad, but it's just, making sure that you're careful in your reading. All right, then let's do this. Unit 8 exam. Oh, you just finished 7, so you guys didn't get to 8. What's up, Elliot? Here to learn. How are you? Sorry you guys weren't able to get to Unit 8 in your class. I do have a video just for that. You should go watch it. It's probably one of the most popular videos on my channel, by the way. Go check that out where I review all the concepts. We're going to do a quick Unit 8 quiz here. 10 questions. When a minnow, you haven't finished 7. Wow. Wow. Sorry that that's happening to you. Yep. I would be interesting to get a poll from all A&P Bio students. What is the last unit your teacher finished? So that would be an interesting poll. I know I've seen the other end of things, but I was in a teacher group at one point, and I saw them talking about how they were just getting to Unit 6 in April. But anyway, here we are. When a minnow is injured by predatory fish, it emits a chemical alarm substance that can be detected by nearby minnows. To test the relationship between this alarm substance and the behavior of minnows, scientists carried out an experiment. First, the scientists exposed minnows in one tank to plain water, minnows to another one, combining these substances. Then okay, good. Yeah, you definitely need to review. There's a lot of 7 in there, but it kind of weeds itself around into all the. We'll go over the FRQs tomorrow, and you see a lot of 7 in the FRQs for sure. Just because evolution is such a pervasive concept in a biology classroom. So here's our graph. Let's go find a thing. Let's just skip straight to the question. Well, maybe this is a long question. Which this may demonstrate a point here. Let's see. We're just going to skip all that and see what we can do. What time tomorrow? 7 p.m. Central. 7 p.m. Central time. Here in Western Kentucky. All right. Which of the following connections between behavior and evolutionary fitness is best supported by the experiment above? When the alarm is present, schooling happens, sheltering happens, time before first capture increases. So all fitness increases when communication's occurring. So I didn't even read all that. It's not hard. Which of the following statements is most likely an explanation for the decrease in sea urchin abundance between 80 and 90? Oh, red tide increases, sea urchins decrease. Oh, this is a whole question. Wow. And you have a decrease in abundance. It's because red tide. What is red tide? During this, which blooms can produce or harm, because the red tide increases and there's more toxins. By density dependent factor, that had a positive effect. No, it is density dependent because it's a biotic factor, right? So it's going to be density dependent that directly cause the environment's carrying capacity to increase. Maybe I'm wrong here. Probably should have read this whole thing. Because this is, sea urchin was reduced by a density independent factor that had a negative effect on embryo development. Yes, I will read through them. It's I'm demonstrating here why it's hurting me not reading through them too. So I'm assuming that I've been reading through them this whole time, so I'm just getting tired. Sorry, we'll do this. All right, the following diagram shows the approximate dry mass in grams per square meter of organisms at the trophic level in the forest. So here we have the primary producers having a ton of dry mass, and then it increases or decreases 90% per trophic level until you get to the top, and you have very little. Which of the following best predicts the effect on the decrease in primary producers' exposure to sunlight? And so if primary producers have a decrease of exposure to sunlight, they're going to produce less biomass, which is going to trickle down throughout the ecosystem. And so each of the trophic levels also going to have a decrease in biomass. Biomass at all trophic levels will decrease. That's exactly what this says. Good. Let's look. Relationships between populations of an ecosystem are often represented using models that show whether a population has a positive or negative effect on the size of another population, and whether this effect is direct or indirect. Common symbols for these types of interactions are shown: positive, negative, direct, indirect. This type of relationship modeling can be done for kelp forest, which are marine ecosystems found primarily along the rocky shorelines of North Pacific. In kelp forest, kelp provides a food source for sea urchins, which in turn provide a sea source for otters. Good. Based on the information, which of the following models best represents the relationship between kelp, sea otters, and urchins? And so we're going to look for a this is a wonky, okay. So sea otters are having a negative effect on sea urchins, correct, because they're eating them. Sea urchins are having a negative effect on kelp because they're eating them. But roundabout, sea otters are having a positive effect on kelp because they're eating the things that are eating them. This is the only one that makes sense to me, because sea otters are regulating the population control of sea urchins. Okay, good. This one doesn't make sense because sea otters are eating sea urchins. They're not positively affecting them. And all the negatives here, we know that's not true because this ecosystem exists, right? If it was all negative all the time, it wouldn't exist. Okay. Human-driven climate change has resulted in shifting weather patterns around the world. For example, in southwestern Australia, rainfall patterns have changed substantially since the beginning of the 20th century. You see that there. Which of the following best predicts the effect that a change in rainfall patterns would have on southwestern ecosystems? Producer levels are going to decrease, and because they're going to have less food available, less production. So let's go find something like that. With less water, species are not adapted to drought conditions will go extinct from the region, disrupting community structures. This is simple, right? There's not more water. Right, we're not getting more water, we're getting less water. Species that live in a region will adapt to sustain drought conditions through the process of natural selection. Not necessarily, right, because adaptation requires a lot of time, and this is not a long time. Species from nearby regions will expand their ranges as the dry ecosystems change to wet ones. We're not seeing that happen. A is the only correct answer. Starting in 1960, a scientist studied a roll of a relatively low abundance predator, the sea star, that Pisaster ochraceus. It's a rocky intertidal ecosystem. To do this, scientists designated an 8-meter stretch of coastline as an experimental area from which all P. ochraceus were removed. The scientists also designated an 8-meter stretch of coastline as control, which were left intact. For the next 10 years, scientists kept those out of that experimental area. During this time, scientists gathered data on species richness from the experimental control. So you have the number of species present in the control area versus the experimental area, right? Because you have this predator this top tier predator that's removed, and what's going to happen? The number of species present is going to decrease, right? Which of the following statements best supported by the information? This is a keystone species, right? We're going to go find that in the answers, something like that. Oh, there it is. Oh, and again, I would have missed this one because I didn't read it carefully. It's a keystone species that strongly influences the structure of its community due to its relatively high abundance. It doesn't have a relatively high abundance. It's not a producer. It's a keystone species whose removal led to the collapse of community and ecosystem structure. This is better. You should read past the first 6 words. The following table lists the number of individual species in an insect community. There they are. Which of the following represents the Simpsons diversity index for this? Okay, so the Simpsons diversity index equation. Okay, this will be fun. This is gonna be fun. All right, so let's find our Simpsons. Let's find our Simpsons equation here. I'm gonna show you real quick. Just as a note, I've never seen a Simpsons problem except for on Con. So it's fine. And so let's look at this Simpsons problem. Diversity index is 1 minus the sum of n over n squared, whereas lowercase n is the total number of organisms and big N is the total number of all species. And so you're going to divide that and square it, and you're going to have a bunch of numbers, and you're going to add all those numbers together, right? And so let's go look at our problem now. Okay. So I'm going to divide 285 by 1237. I'll pull up my calculator here. You won't be able to see it. But let's see. 285 divided by 1237 is 0.23, and I'm going to square that. And that's going to be 0.05. Let's say 0.05, right? So we're going to keep 0.05 in our mind. I'm actually going to pull up another document here. 0.05. I don't have any scratch paper, so sorry. And then 175 divided by 1237. Square that. I'm going to get 0.02, right? So I'm just adding these together. 362 divided by 1237. And we're going to square that, and we're going to get 0.085, and so we're going to go ahead and just say 0.09, since we're rounding to the nearest hundredth. And then 415 divided by 1237 squared is going to be 0.11. And so 0.11 plus 0.09 is 0.2. 0.2 plus 0.02 and plus 0.05 is 0.27. And there it is in black and white. And so this is just plug and chug, right? It's not asking you to do anything with this number. This is actually wrong. What I was about to miss this, because Simpson's diversity index is 1 minus this number, which would be 0.73, right? It's about to do all that math for nothing, but I miss it. The following table shows the internal body temperatures of a polar bear during various activities. There they are. They're pretty much all the same. Which of the following statements is most likely true about polar bear described? Polar bear's body temperature remains relatively constant due to thermal energy generated by metabolism. And that's just correct. But let's go look at the other things. Is ectothermic, using internal mechanisms to maintain body temperature. Ectothermic and maintaining internal mechanisms are not the same. Is endothermic, carrying out certain behaviors in order to maintain a constant. This is also mixing definitions. Polar's body temperature increases significantly. It did not. So A is the correct answer. Population of deer in a forest is at a maximum per capita growth rate of Rmax at 0.4. The population size is limited by the carrying capacity of 900, which is K. Which of the following is the growth rate of a population of deer when the population is made up of 300 individuals, which is N. Okay, good. So we have 900, 300.4. This is another math problem. And so one of the things I've told my students is that I have not seen any ecology math on the AP exam in years. And so we might see it. I'm not expecting to see it, right? So we're going to do, and this over here is the rate. This is a change over time, right? So Rmax was 0.4, N was 300, and 900 was our carrying capacity. So we're just going to do some quick math here. 900 minus 300 is 600 divided by 900, which is going to be 0.66, and we're going to multiply that times 300 and then times 0.4, 80. What is endothermic and the other thermic? Okay, good. Endothermic is controlling temperature from the inside. So endo means inside. So it's like us, right? We control our body temperatures from within, whereas ectothermic has to control their body temperatures from without, like a snake, something like that, has to do certain behaviors in order to change its body temperature. And so though that last question was mixing the word with the definition. It was exchanging them. There it is. 80. Oh, this has a calculator. Well, I'll do that next time so you guys can see my calculations. Probably won't have it next time though. The following tables describe species that make up two forest tree communities. So we got this. We're going to have it next time. Which community is likely to be more resistant to changes in its environment and why? It's because this one is going to be more stable. There's more diversity here than there is here. Basswood would be really small. If something happened to it, it would really affect it. No, that's really hard. All right, so we are at the point in the evening when I have completely... Unit 9? What is Unit 9? Explore biology through simulations. You know what? I'm going to leave you to it. You guys can do that on your own. So tomorrow evening, I am going to be here at around 7 p.m. Central Time in order to go over some FRQs with you guys. For instance, I'm going to be going over this FRQ, 2024 FRQ. Yes, so we went over Unit 7, and so you could go back and you could watch that, or last question, do you think there will be the cycles? Oh, you're talking about... There's not a Unit 9, by the way. That's why I was laughing. The cycles you're talking about... This, like nutrient cycling, I think. Yeah, 8 p.m. Eastern, correct. There could be questions about these nutrient cycles. My guess is that the questions are not going to be over these individual words here, because they're not in the course description really. But my guess is that Krebs and glycolysis, yeah, those will definitely be on there. And we'll cover some of that in the FRQ time tomorrow. And tomorrow, if you have content questions, I'll be able to go over them as well. But my guess is that questions about the cycles will be like, why is nitrogen necessary? Where is nitrogen found? that kind of thing. And so, as opposed to like define nitrification versus assimilation or anything like that. I don't think that's going to be on there. Yeah, no, these you will definitely have this stuff on here for sure, and we'll go over some of this tomorrow if there are other questions. I can see more people showing up, but and maybe I should have started later. I don't know, but if I started later, I would be worth very little. And so tomorrow evening, I will be starting around 7 p.m. Central, and I'll be going over FRQs and any content questions that you guys have that are related to that. Thank you guys for coming out. It's been great. I always enjoy this sort of thing. Did see one of my own students in here, and that was just kind of cool. But a lot of you guys I'll never meet, which is also really cool. Yeah. If you have questions, put them in the comment in the video, and I'll try to answer